So thanks very much for the invitation to be here, and excited to share some of our results with you today. As Tom said, I'm going to talk about periprostatic adipose tissue prostate cancer, and a couple of co-investigators here in the room. So by way of background, oh, and I see I don't have my my animations. I'll give you get it all at the same time. So we know that obesity is a risk factor for advanced prostate cancer, one of a dozen tumor organ sites as being related to obesity, and we think that visceral adiposity is a driver of the association, with visceral fat being the more metabolically active, potentially harmful fat. And just as a by way of introduction, I'm showing you here the CTs. So these are transverse sections through the abdomen at the level of the belly button to illustrate vast differences in adipose tissue distribution for men, even of similar surrogate measures of obesity, like waist circumference and BMI. So, for example, the man on the right there has a lot more fat around the organs, a lot more abdominal visceral fat relative to the man on the right, even though they have similar waist circumferences. So we've known this for a while. But what we're interested in this project is in periprostatic adipose tissue. So what is that, and why are we interested? Well, this is a fat depot within the pelvis, and so you can see the top CT image there on the right just shows you that line indicating that region of the body, the pelvis, where the prostate lies. And so you can see then in the middle, this is the CT transverse section through the pelvis. So you can see the hip bones either side, the prostate in red there in the middle. And this is a density threshold that's been applied to the CT to highlight the adipose tissue within the pelvic cavity there in green. And then at the bottom, I've just given you a 3D sort of reconstruction of the fat type that we're interested in. So the prostate in pink, and then the green being the adipose tissue surrounding it. So like some other organs in the body, the prostate also has organ-associated fat and visceral adipose tissues surrounding the organ. And we think that we're interested in it for a couple different reasons. I suppose there's been interest in studying periprostatic adipose tissue over the last decade. For example, a number of studies in the lab have co-cultured adipose tissue explants from, say, men undergoing surgery for prostate cancer, taking samples of that adipose tissue surrounding the prostate and co-culturing of tumor cells in the lab, finding that it promotes tumor cell proliferation and aggressiveness in vitro. And then in parallel, other studies using imaging obtained for medical purposes to quantify the this periprostatic adipose tissue, which you can't really measure any other way, and finding that it seems to correlate with higher tumor aggressiveness. But I think the compelling thing for me is that there's some pronounced gaps in the research to date. So the published studies kind of broadly fall into one of two groups, and these are the studies that have used imaging to quantify PPAT in people. And that they're either performed, because of the need for medical imaging to quantify this fat, they're either performed in men who are undergoing, say, radiation therapy for prostate cancer, so they're getting imaging to plan that treatment, or perhaps men undergoing that sort of diagnostic pipeline for prostate cancer, so they're having imaging for that purpose. So they're either performed, these studies, in men with or under suspicion of having prostate cancer. Prospective studies are completely non-existent in this space. And in addition to that, the studies that have used imaging to quantify PPAT tend to report various different definitions of this adipose tissue depot. And then finally, because of the need for manual effort to contour and measure and outline the adipose tissue depot, these studies have been limited to several hundred individuals, say, in size. So with those things in mind, we proposed and were funded by the World Cancer Research Fund in 2023 to perform the first prospective study of periprostatic adipose tissue and aggressive prostate cancer risk, hypothesizing that maybe we could provide a little bit of preliminary evidence to show whether, or to test whether PPAP may be potentially modifiable through lifestyle changes, for example. And then secondly, hypothesizing that it may be causally related to risk of aggressive prostate cancer. And then the little figure on the right shows sort of broadly in brief our approach, that we're using data from 2 longitudinal studies with imaging in cancer-free study participants, UK Biobank, which we've heard a lot about today already, and then another longitudinal study based in Iceland called Aegis Reykjavik. So using imaging in these studies to derive a robust measure of PPAT in men without prostate cancer for the first time, and then to use complementary approaches, I guess I have timings or something, so I'm behind, to a traditional observational epidemiology approach to look at the association with prostate cancer risk, and then in parallel a GWAS and MR analysis. So maybe we don't need this slide because many are familiar with the UK Biobank, but just to give you a quick overview of the study design that enables us to study this prospectively. And I'll just focus on UK Biobank for today because we don't have the data from the Aegis Reykjavik yet. But about 250,000 men recruited between 2006 and 2010. And then the imaging study, which is what we need to measure the PPAT, starting in 2014. And I think just recently they have completed imaging now in 100,000 members or participants in UK Biobank. But at the time, 2 years ago, when we accessed the imaging, there were whole body MRs for just over 33,000 men without prostate cancer. And then I wanted just on this slide as well, just to give you our definition of PPAT. And so you can see our 3D sort of model up in the top right there, the prostate in green, and then the adipose tissue around it in brown. And we decided to call PPAT the adipose tissue that was most in closest proximity to the prostate, so up there labeled as PPAT. And there's also adipose tissue more in the posterior region of the pelvis, more surrounding the rectum, which we're calling PRAT, or perirectal adipose tissue. So that was our first challenge, was how are we going to quantify these adipose tissue depots in this large number of men? And so we had to do it through AI. And so these were co-investigators at Trinity College in Dublin who developed trained an NN-UNet model by inputting manual contours, train the model using these whole body MRs from UK Biobank. These scans were obviously huge in size, and so they developed like an in-house code called Unpack to just pull the relevant MR slices from the pelvis, so that the model could contour PPAT in these what turned out to be just over 28,000 men with imaging that we could analyze. And then they further applied PyRadiomics, which is an open source software to extract radiomics or quantitative features from these images. So I'm showing you here the data from the men without prostate cancer in UK Biobank, and you can see up on the right there, that's the distribution of the periprostatic adipose tissue. So you can see the median amount is just under 14 mils, so not very much of it. And then here is how it relates to other more well-characterized adipose tissue depots. So I'm showing you here, the first column there is the periprostatic adipose tissue. And you can see that the correlation is stronger with abdominal visceral adipose tissue compared to BMI, for example. And so broadly, we see that the PPAT correlates more strongly with other visceral measures, which I suppose makes sense because we think of it as a visceral fat depot. But I suppose the other thing to note is that, you know, it's a moderate correlation, and it's certainly, you know, we may not be able to infer the amount of PPAT by using other surrogates very well. And so next we thought, well, I wonder, you know, how does the amount of periprostatic adipose tissue relate to measures of systemic metabolic health? And so here I'm showing you the Spearman correlation coefficients with blood measures that were taken from blood samples that participants provided a baseline in UK Biobank. So you can see when you compare the relationships for abdominal visceral adipose tissue with PPAT, that we see similar patterns of correlation, although they're stronger really for the abdominal measure of visceral adipose tissue compared to PPAT. Given that PPAT was moderately correlated with abdominal visceral adipose tissue, we kind of wondered, oh, well, is it just because PPAT is correlated with VAT that, you know, that's why we're seeing this pattern of same pattern of relationship. And we used partial correlation to adjust for abdominal VAT and found that the relationship of PPAT with these systemic measures of metabolic health was really attenuated. So I suppose we think that the effect possibly of PPAT on the prostate is quite likely to be local. And I suppose it makes sense if you think even just in terms of the quantities of these adipose tissue depots in the body. So, for example, the average man in UK Biobank has about 5 litres, of visceral fat in their abdomens, as opposed to a mere tablespoon of periprostatic fat. So from here, then we went on to have a look at the relationship of adiposity measures with participant characteristics. And so again, that this was the first time that this has really been done on a large scale in men without prostate cancer. And so as we, I see it's flying ahead of me now, but I wanted to draw your attention to the, sorry, maybe I shouldn't interfere, to the age and race relationships of PPAT and other, you know, with other adiposity measures for comparison here. So these are the beta coefficients from linear regressions of these standardized adiposity measures. And so you can see that, I suppose we were struck by the pronounced relationship of PPAT with age that we see about that men in the oldest age category of UK Biobank have twice as much periprostatic adipose tissue as the men in the youngest age groups. We see this to some extent for PRAT, for perirectal adipose tissue, and for abdominal visceral adipose tissue, but not to the same extent. And of course, we know you know, from other published work that visceral adiposity tends to increase with age. But again, for the abdominal measure of visceral adiposity, it's really in the region of a 20% increase with age, whereas PPAT, you know, we see this to be more pronounced. And I suppose we know as well that BMI is not a great surrogate of obesity with age. And indeed, we see that the oldest group of UK Biobank participants actually have lower BMI than the youngest. We also know, or it's been established, that there are race differences in adipose tissue distribution. And we see these again for PPAT, as we do say for abdominal visceral adipose tissue. Most pronounced for abdominal visceral adipose tissue. For example, you can see that Black men have roughly 50% less visceral adiposity in their abdomen compared to white men. And we can see that this is sort of the same patterns of association for PPAT, although slightly less pronounced. While in contrast, Black men actually have a higher, 20% higher BMI than white men. So pronounced age and race differences in adipose tissue. So adjusting then these models for age and race, we looked at other participant characteristics associated with PPAT volume. And again, we see sort of overall similar relationships for PPAT as well as for abdominal visceral adiposity. For example, that men in the highest education category tend to have lower amounts of PPAT and abdominal visceral adiposity compared to those who only complete some of secondary school education. Smokers and daily alcohol drinkers tended to have more of these visceral measures of adiposity. Men with a history of diabetes, which was very much higher abdominal visceral fat, which and so similar, although less strong relationship with PPAT volume, and that men reporting the highest amounts of physical activity tended to have lower amounts of both of these adipose tissue depots. And I suppose one difference that we see between these two graphs is that we see that men with a history of BPH or benign prostatic hyperplasia had a 9% greater volume of PPAT. And we sort of thought, well, maybe PPAP could have a role in non-cancer prostate conditions too. And indeed, we know from some of those in vitro investigations with co-culture of PPAP explants with cells that it promotes proliferation. So I'm afraid this slide has a thing. Oh, you're not going to be able to see now that. But I wanted to end by showing you some of our predictive analysis using the radiomics features. So I had mentioned a few slides ago that we used radiomics to extract those radiomics features or quantitative features of imaging, because we wanted to see whether there were sort of morphological features of PPAP that could be predictive of prostate cancer risk. And so this analysis was done by creating a nested case control study within UK Biobank, finding the 297 incident prostate cancers that were diagnosed post-imaging, and then age matching those to 297 controls. Applying the radiomics software to identify our features, doing some dimensionality reduction on those, and then just logistic regression and an ROC analysis to find those features predictive of prostate cancer risk. And of course, here you'll know that we're just looking at overall prostate cancer risk without having a grading stage in UK Biobank. What we did find, and what luckily you can see, is that we did find 31 significant features of PPAP that were predictive of prostate cancer risk. And I'm showing you the shape features just on the right there. They're sort of the most easily biologically interpretable features. And so you can see features around diameter, volume, and sphericity, surface area that we thought were interesting. And when you put these features together in the model, it produces an area under the curve of 0.64, which is sort of in the region of what PSA can do. So we think this is, a first step in giving us some preliminary evidence that features of PPAT, including those around the size and shape of this adipose tissue depot, might be important in the risk of prostate cancer. And so next steps, so we're about halfway through the project. We're working with Bristol colleagues on a GWAS and MR for this fat depot, and then a traditional observational cohort analysis in UK Biobank and the Aegis Reykjavik study. So I'll leave you there. That the few takeaways that I feel like we have so far are that BMI and other adiposity measures and surrogates, aren't very good surrogates for PPAT, which really needs to be measured on imaging to understand it better. That we hypothesize, I suppose, that the effects on the PPAT are most likely local and not systemic. We see an interesting relationship of PPAT with advancing age that we'd like to look into more, and that we have preliminary evidence that maybe there are some aspects of this fat depot that could be important for prostate cancer risk. And so then just to finish by acknowledging the co-investigators on the grant, the team in Trinity College who produced the AI model, measured the fat depots, in Bristol who were doing the GWAS and MR, as I mentioned. Co-investigators at Oxford who've really provided a lot of support with UK Biobank data analysis, as well as our PI of the Aegis Reykjavik study, and of course, thanks to WCRF for funding our work. And I'd be happy to take any questions.